Welcome, welcome everybody. Welcome to the Netherlands. Welcome to the country that will host the IUPAC World Chemistry Conference and General Assembly in 2 years from now. We hope to get to meet you, all of you, in The Hague in August 2023. Take also a look at the website www.iupac2023.org and see what you can expect in that time. And hopefully it will be a conference without a pandemic at that point. So let's start today's program. My name is Timothy Noël. I'm a professor at the University of Amsterdam, and I will be today's moderator. Today we have a panel discussion that is part of the World Chemistry Leadership Meeting about the future of chemistry in a world of AI. We have several themes we would like to discuss regarding artificial intelligence, and especially we want to figure out how will the development of AI influence chemistry in the future. For each theme, I will provide a short summary that will end with a statement, and then we will discuss with our panelists and about that statement and they will give their opinion on the topic. And after that, we can also discuss with each other about the topic itself. After a brief discussion part, participant questions via the Q&A can be asked to the panelists. As a participant, you can find the Q&A button in the Zoom bar at the bottom of or on the top of your screen. On behalf of time management, we will ask the questions that most people are interested in. So you can upvote other person's questions in the Q&A by clicking on the thumbs up button. So after that, the most relevant questions will be presented on top, and then we can ask those questions first. After approximately 20-minute discussion per team, we will continue on the next topic. And at the end, we will have some time for remaining questions from the participants. With that housekeeping, I would like to also invite the different panelists to have the panel discussion. We have selected 4 experts from both industry and academia. And it's my honor to welcome Dr. Ola Inquist, who is head of molecular AI in discovery sciences at AstraZeneca R&D. Also, Dr. Barbara Zdrazil from the University of Vienna, Dr. Hugo Ceulemans, Scientific Director, Discovery Data Sciences at Janssen, and Professor Maarten from the University of Maastricht. So welcome to all of you, and please introduce yourself briefly. I would like to start with Dr. Ola Inquist.
Okay, hello, my name is Ola Inquist. So I'm at AstraZeneca, based at the site in Gothenburg in Sweden. So I'm an original computational chemist. So I made my PhD in that subject in Lund. Then after postdoc and working for 2 small biotech companies, I joined AstraZeneca in 2004 as a computational chemist, and then successively first leading a team and now a department, successfully also changing tasks from starting with more traditional computational chemistry, moving over to more and more machine learning and AI, and now it's the main focus of my work. And it's focusing on applying machine learning and AI to drug design. Basically, how can we generate molecular ideas? How can we predict synthetic routes? And how can we predict the properties of the suggested molecules? And it's both around the science to develop new methods there, to productionalize the science, to have a robust solution. And you can see also we have released most of them as open source, and then to apply it on projects. And since I come from big pharma, it's so to say a lot around the portfolio. It's not only to apply it in one project here and another project there. It's really to move the needle for the whole project portfolio with machine learning and AI, to be able to reach clinical candidates faster than that, so to say, will cost benefit the patients, that will be faster product to the market.
Thank you, Ola. Barbara, can you introduce yourself now?
Yes, of course. And first of all, thanks for the invitation. I'm looking forward to the nice discussion today. So my name is Barbara. I'm working at the University of Vienna as a group leader. I'm also now working for EBI in Cambridge. for the Open Targets project. And in my role as an academic and group leader at the University of Vienna, we mainly focus on a data-driven molecular design approaches. And what does this mean? So it means that from the beginning of a drug discovery question, we always try to include data-driven approaches. So we base our decisions really on the data. Technically, this means that we generate automated workflows that can integrate data, analyze data, curate the data. And it's also very important that we always try to make these workflows available to the community. So that's a very important point as well. And in terms of modeling, we also try to automatize modeling to a large extent. So we do, of course, machine learning, but we also combine it with structure-based modeling. So target-wise in my group, we are mainly interested or focusing on off-targets like transporters. So it started already in my PhD, actually, where I worked on P-glycoprotein and other ABC transporters. But now I'm more focusing on SLC uptake transporters, which are very important because they can lead to drug-drug interactions, liver toxicity, for instance, like organic anion transporting polypeptides, organic cation transporters. So target-wise, I'm really more interested in trying to understand targets that we should avoid to target in drug discovery rather than real drug targets. And at EMBL EBI, mainly I'm contributing, as I said, to Open Targets, which is a project to systematically identify and prioritize drug targets. And also here, I'm more contributing to the safety aspects of the targets. Yes, and as I said, I'm very glad to be here, and I'm looking forward to an interesting discussion.
Thank you, Barbara. Hugo, can you introduce yourself?
Yes, happy to. So my name is Hugo Ceulemans, and so first of all, also thank you for the invitation to be here. I started out long ago as a physician, turned molecular biologist and bioinformatician, and in that capacity, I joined Janssen in 2008. Slowly, I've been moving closer to the interface of bioinformatics and chemoinformatics, predicting the properties of compounds, understanding the properties of compounds, but also making headway on how to make such molecules. AI/ML is the ideal setting to combine all those flows. We deploy those insights, those methods across the portfolio to progress those different projects. And we have a healthy interest in identifying where are we missing certain data types, certain data sources, what are the gaps, where are we not strong enough to help support the portfolio projects, and how can we remedy that? So how can we pull in those data? Looking forward to a productive discussion.
Thank you. And last but not least, Maarten, can you introduce yourself?
Yeah, thanks, Tim. And thanks for inviting me to join this interesting panel. My name is Maarten Honing. I'm an analytical scientist, chemist, you could also say it. I have a special interest in what I call system imaging. And that means for me the monitoring of time resolved and molecular structure resolved processes. And especially, like Hugo said, also understand what are the basics there and what do we miss as analytical scientists, because we have a lot of sensors, a lot of methods, but I dare to say always that we miss 80% of the molecular information we are looking for in our samples. I have a background in industry for a long time. Before 4 years ago, joining Maastricht University, I've been working in pharma for nearly 20 years, supporting medicinal chemists, toxicologists, pharmacists, and process chemists in process development, structure-based drug design, and all those aspects. And at DSM, a big chemical company at that time still, I was also linked to industrial chemistry, caprolactam, melamine, and also material sciences, applying analytical chemistry to understand processes, properties of materials. And that's really my cup of tea. I'm very fond on chemometrics and analytical data quality, statistics, and so on. So I absolutely see an added value of AI, and I'm interested also to apply it in our research. Lastly, to say, not only to investigate medicinal chemistry or target ligand interaction, but apply analytical artificial intelligence, improving analytical methods in the analytical laboratory. So thank you.
Thank you, Maarten. And with that, we will go to our first presentation, I will make a short summary of that. And it's actually by Dr. Andreas Bender from the University of Cambridge. And his talk was about the processes and tools of the future. And so in his talk, he was describing that processes really grow out of technology, but also costs. And that's like even a push factor. And then really the beliefs also in the company. But then he said something about that actually, if we want to change a process, it's very hard, partially because of human resistance. I mean, people do not like to change too much. And then also the entanglement of different parts with each other. So if you want to change one part, you essentially have to change the entire part, and that creates also a resistance. But he also said that disruption is on the way, and especially for data-first companies where processes are built from scratch. That's the easiest, because then you start with a clean slate, and then you can, well, do it essentially as you want to do it. And he says you have to really rethink the entire process to maximize the efficiency of the new capabilities that is given by AI. And so his suggestion for drug discovery was that to start with the data-driven starting point and optimization based on that data using machine learning, and then also actually add there the use of expert knowledge. The problem so far, he says, is that limitations are in data, both quantity but also predictivity. So the question that we need to ask ourselves is, what do we need to measure for a specific question? So it's actually very important to ask the right question and then make sure that you have the right data to be able to answer that question. So his conclusion was that with the tools at hand, we need to change the processes, but we should not support existing processes with AI, but rather start from scratch. And so the statement that we would like to discuss today is, can old processes per definition can never be useful when combined with new AI technologies? It's a little bit like a supercomputer built in an old timer. So you need to really rethink the entire process. So that was his message. And you will also see there in the screen what the statement was that we will discuss. And maybe I would like to hand this question first over to Ola, and maybe he can kick off the discussion.
Okay, yeah, absolutely. Thanks a lot. And yeah, so with this statement, I think I disagree. And a main reason is of course, because if you apply that while I'm working in drug design, applying machine learning AI for drug design, so the way to do this would be start a new company that can start from scratch with the new processes. Clearly, since I'm in a larger, older company, I have a different viewpoint, and I do think you can change processes. I mean, it's a challenge. I mean, it's like constructing and driving a car at the same time. But I do think there's actually a lot of learning between the old and new processes. And I think we have seen that in several cases, that it's actually the way one has worked traditional can very much inform a new way of working, where you more emphasize machine learning and AI. So it's very much a process. And I think the end goal should be that the machine learning AI is much more in the center than it's been historical. But I think you can have it as a continuous process, not necessarily as a discontinuous process, as is indicated in the statement.
Could you give one example from your own experience that you can reveal, of course, or like something generic?
It's like you can, for instance, apply new methods in so to say, in a traditional drug design setting where you have the design meeting. It's not that you really need to have potential specific meetings for just the AI design. You can basically train the computational chemists that will be part of those design meetings, that besides using the standard tool they have been using before, also starts to use machine learning and AI tool. And you can propose AI-based molecules alongside molecules that have been proposed with more traditional medchem thinking.
And how is the rate of discovery? Do you have a ballpark figure, like compared to the old-fashioned way and now the new AI-enabled one?
How do you mean with the rate?
So yeah, one of the things is that you want to go faster to the solution, right? So
Yeah.
how do you see that within your company?
I think it's fair that we don't have that all the numbers for that, because you need to apply it on a portfolio perspective.
Right.
There are also confounding factors that the target portfolio, I think in pharma, is changing at the moment due to progress in the GWAS studies, in functional genomics. There are different types of targets coming in compared to what we've been working on historically that might have been more focused on certain target classes. So I think it's the measurement problem is to have a fully objective is very tricky. So what we're usually talking about is like real world evidence, just what you have for a drug on the market. I mean, is the uptake in the projects? Are the compounds that you have proposed with MI and machine learning, are they going for synthesis and so on? Are they on the critical path to deliver the compounds that will be taken from milestone transition, say a clinical candidate and so on? So it's a lot around real world evidence. To have an absolute number that method A is better than method B, I think is very difficult because there's a lot of confounding factor. I mean, for me, the goal is like to have machine learning and AI for drug design. You see it as the same way you see crystallography. I mean, it's obvious if you can solve a crystal structure with the so to say compounds from your series in the protein, you do it because you know that will be useful information. I hope it will be the same with machine learning and AI. That is so obvious, just like crystal structure, that it will be default.
Yeah. Maybe Hugo from Janssen, maybe you can tune in here and give your perspective.
Yeah, by and large, I concur with Ola. So if you were hoping for a discussion, this is not the right point. And what I do like is the analogy with cars, and particularly with self-driving cars. I think the process is very similar. So in both cases, we've been working intelligence already for decades. If you today would go to the highest end, best supported car, you can still not have it self-drive on the public road. At the same time, the lowest end cars would already have some intelligence in. So it's not a question of do you do intelligence, it is there. The extent to which is very different. Also the experience how your cars start having more and more sensors. Initially you see more complexity, more data coming in. Then the car manufacturers start whittling that down. It becomes more intuitive. It's not a completely painless process, but people who are getting used to more and more, we are all driving cars that have these gadgets in them, and we are getting used to them. I think the process in AI/ML augmented, it's the same way. So we bring in more information. We try new ways of sharing that with the colleagues. Some of them work less. Slowly, they also become more accustomed. Hey, we put the brake on, but we start to understand that sometimes is useful. Here, we accelerate. We wouldn't have done that, but it seems to be a good hunch. There becomes more comfort also because more and more younger people that are more accustomed to these methods or joining the ranks. So it's a gradual process, and we don't believe in that complete holdover. It is more like a Werner von Braun thing. If you want to do a space program, you build it up stage by stage. You deal with everything, you bring it in, show that it works, bringing the next thing, raising the comfort level.
Right.
So all in all, pretty close to what Ola referred to.
Barbara, maybe you can tune in from the academic side. Yeah.
Exactly. So yeah, I really agree with what Ola and Hugo already said, but I think it's very important that we distinguish between all the new methods and all the new processes. And I think what Andreas Bender meant in his talk was more about, okay, there are all the new methods, but if we want to include new methods, we have to rethink the whole process. And in this respect, I agree with Andreas, because I think basically the point he made was we have to make data a basis for all our decisions early on. And I think that's the main difference, other than okay, bringing in more new method will, as also Ola and Hugo already said, is an iterative process, right? We have to learn within the process what works and what doesn't work. But really the process itself, I think, needs a change. And this means we have to rethink completely. We always have to make data the basis for our decisions in drug discovery. And we can do so now, right? Because we have all these different data sources. And I think we will talk about this a bit later. We have these knowledge graphs. We can really leverage all this data. And I think that's the main difference. I don't see so much of a problem to include new methods. And with respect to older methods, I think we also have to define what do we mean by old and new, right? I think talking about methods, some of the older methods are still very valid. And especially in this now age of AI, we have to think about simpler methods sometimes. So now we tend to use those very complicated algorithms, but then they're less interpretable maybe, right? So we should also use the simplest method possible. So that's also something I think we have to consider. But in general, I agree, it's an iterative process. We go step by step. It's not really completely redesigning everything, but the process per se, I think we really have to re-engineer this. And in this respect, I agree with Andreas Bender, what he said in his talk.
Maarten, your opinion?
Very interesting, because I misunderstood the word old processes, and I was thinking of production processes. I can come back to that. But truly so, I'm not agreeing with the statement. And yes, as a technologist, analytical technologist, I'm very well aware that when you bring technology to a medicinal chemist or to a pharmacologist, that they are very awaiting, I would say, and just looking what the technology will bring them before they start to believe. And on the other hand, and that's natural behavior all over industry, all over sciences in general, I would say, and you can make a lot of mistakes, because if you promise too much, then you lose quicker the confidence than you tried to build it before. On the other hand, for example, in chemical industry, and especially the big industry, they have hundreds of years, sometimes tens of years of data on their processes, and nobody have been looking from an understanding point of view to those data, and they are very valuable. And I've been working at the Camelot site here in the south in Limburg, and there's, for example, maintenance data of the factories for nearly 50 years of thousands of sensors. And there's a lot of information there on predictive maintenance, and that's also a new field there. I visited one time a champagne company, and they had 120 years of information in books about the temperatures on and the type of grapes they grow. So if you want to show the validity of your technology, there's old data, and you can show the validity of your approach and the added value to your new colleagues on basis of all data, not only for new data to be built. And of course, you have to build it up in your organization like that. So I disagree a little bit. On the other hand, again, together with Hugo, you cannot use very artificial advanced systems when the data are not complete enough. You need sensors to do so. But I think there's a lot of data out there, high quality data in the field, enough to prove the validity, and especially like Barbara said, also the validity of different approaches, either machine learning or artificial network, neural network approaches to show how important artificial intelligence will be in prediction, and especially in prediction of new processes.
Right, and Hugo also said something important, is that with the new generations that are educated a little bit differently, it might be easier to implement these new changes. So I think this is also an important thing, that if machine learning and AI is going to get more traction, then probably there will be some courses also in classical chemical engineering, but also chemistry and across other disciplines. And I think this might be a good strategy, and probably also will have more impact than later on. And Ola, you want to say something about this?
No, just reflecting on old process, more thinking about the experimental processes, that I think it's in some area the experimental throughput is going forward very quickly, particularly in the omics area and next generation sequencing. So I think due to the enormous increase, or I should say exponential decrease of sequencing cost. I mean, old processes are per definition outdated also from a data science AI perspective. Now, when you can generate enormous data for next data sets for next generation sequencing, yeah, you need new processes for data storage, the data loading and the data integration and data analysis, because the experimental techniques are progressing so quickly. So you need simply completely new processes for the data science and AI part.
Yeah, and I would even add also maybe the discussion also on quality. So you can compare in medicinal chemistry a lot of molecules on basis of the structure and activity, but if the activity assays have a standard deviation of 35%, then it's extremely difficult, of course, to really pick out real correlation steps there. So I would say the multimodal approach, so the amount and the relevance, and that's also what Hugo says, the relevance of the data linked to the process which you want to investigate is so important. So it should be also recurring, where AI helps you to redesign your experiments or redesign your analytical sensor technology in order to find really those chemical or physical actors which are the crucial ones who drive your process or your biological process. So we have to make a lot of steps. And again, I've seen also in omics a lot of blah, 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 I would say, a lot of correlation, but hardly any causality between molecular systems and networks. And that's where we have to go in order to create understanding, and from understanding really predict what's going to come to us in the future.
Here too, we can refer to that analogy of the self-driving car. We are now packing those with sensors. I mean, that measure things every millisecond at an accuracy that is unprecedented. At the same time, you cannot overwhelm your end user, the driver, with all of that information. So you need layers to digest that information, to present it in a workable way. Also, to the extent, are we not changing? So to Barbara, I completely agree. We have to revisit the process, but it's not that it cannot be combined. It will be combined gradually. And we're also seeing that in those cars. You see those Teslas where they try, well, can we pack more information just by making a bigger screen? But then it's in the middle of that car. Is that working? Have the impression that we're moving away from that. In the same vogue, we see that as we capture more and more information, we digest it to something usable. But we also need to experiment with what does work. Can we provide more data, or should we reduce it in a more clever way? And we see that all those decisions are actually slowly seeping through in car design. Cars are going to change as a result of this process, just like the pharmaceutical industry, the chemical industry is going to be impacted slowly by this interaction of the end users with the information and the way that the insights are being presented.
Mm-hmm.
And
yeah, Barbara.
Yeah, there's just one more thing I wanted to add. I think we shouldn't forget about this. So when we think about expert knowledge, right, which clearly is like an old process, to include more expert knowledge or intuition, right, as a basis for decision. And that's very much a topic, I think, in organic chemistry, especially, right? What to synthesize next is an old question that is still there, even though we have all these possibilities now to use machine learning to help us take those decisions. So I think we really shouldn't forget about old methods or old processes and always still include them, especially really those decisions that are somehow related to our gut feeling, maybe, especially in organic chemistry, I would say. So not to forget about this as well.
We also got a question from Egon Willighagen, who is at the University of Maastricht, if I'm not mistaken. And he asked, actually, would the actual collection of data not be an old process? The only innovation in the past 20 years is FAIR, which is findable accessible, interoperable, and reusable, and is open same for validation. Is that not a quite old process by now? And is there maybe a better way of doing collection of data? Does anybody have an opinion on that? And this question.
Maybe I can add something.
Yeah,
I think there is one difference is in many cases the volume of data. I think that the term big data, actually the definition is that the data in itself is a problem, because to make a high performing pipeline for analysis, if you have say so to say omics data and so on, it's something new, and that's a field in data science in itself. I think that's also an understanding and apprehension for data more now. That is, you have of course the claim that it's the new oil, and that of course debatable, because if you want to be compared to oil or something like that, but there is, I think, a more people are valuing data much more. So people are going much more back and look in electronic lab notebooks. Can we actually programmatically extract the data, say from medchem ELN or biology ELN? So there is a much more appreciation of the value of the data now compared to definitely 10 years ago and maybe even 5 years ago.
And maybe if
maybe Barbara, you go first, and then Hugo.
Okay, sorry. So maybe to add to this, I think what Egon might refer to as well is, I think that publishers, of course, have a responsibility here, right, to publish data according to fair standards, right? And also myself, I'm also an editor, associate editor of Journal of Cheminformatics. That's one of the main goals we have, right, to make code and data available. So I think, yes, we are still a bit stuck in old processes here to how we collect data, especially when it comes to publishing, because chemistry data isn't yet machine readable in papers. And I think that's a big goal that we have, but we are really in the very beginnings, because it's really difficult to force people to submit the data in that form. Yeah, but I think that's a very important point to make. And I think we'll have a few more discussions around data quality later and how to make data fair and open. So maybe we keep that a little bit for later. But I really just wanted to emphasize it's also a responsibility of every scientist, but also of publishers and journals to help this process to make data fair.
Hugo, you wanted to say something?
I agree with the importance of fairness and openness, as mentioned by Egon and commented on by Barbara. I disagree with the statement from Egon that is the only innovation in the past 20 years. We are currently looking at how can we better represent chemistry in an explosively more diverse way than we have done for decades. So over the last few years, the ways that we're looking at it from a quantum perspective, from different representations, in essence, every machine learning is an attempt to re-represent the data in a more relevant way. So we will definitely come back to that, but that fair and openness would be the only innovation in data over the last 20 years. And I very much disagree with that statement, and I hope to be able to convince Egon together with the colleagues of the contrary.
Maarten, you have any opinion still on this?
No, I had nothing to add. Also, completely support what Hugo has been saying.
Okay, I think that's then our first discussion, and we will go to the next one, which was it's a little bit different. It's about crop science this time, by Wendy Paulson. So she gave a couple of examples if you've seen the presentation, and I will just highlight 3 that I liked. One was the use of sensors on a tractor and then use a drone to better register what is going on the field actually. And so then they use statistical models to track which compounds have a better performance. And so the whole idea was to eradicate subjective human assessment, which allows faster and more accurate decision making. The second one was also an interesting one, which was a sensor on a beehive to measure the activity of bees. And in that way, you can evaluate how new herbicides, insecticides, and fungicides are affected on those, and what are the actually what's going on with the bees at that point. And so before a beekeeper would assess this by just monitoring his or her bees, and that puts a lot of subjectiveness in the equations. And so with these new AI methods, much more precise measures can be made and much more unsubjective conclusions can be drawn. And then the last one that I would like to point out was that weed control products are currently sprayed on the entire field, on every crop. However, by using AI, one can detect weeds and microspray locally where it's actually needed, using, for example, spray drones. Everything is robotized, less manual handling, less compounds on the crops, and that means a safer overall process, and also safer for the farmer, safer for the environment, and safer for the crops itself. So the conclusion that Ms. Wendy Paulson from Biocrop Science made was that smarter farming can be done using AI, and it also minimizes then actually the importance of experience in farming. Would AI be able to make of everyone a decent farmer? That was one of the questions I had for myself. And so the statement that we would like to discuss is, in agriculture, the development of AI-driven or oriented technologies are more important than practical or hands-on tools and techniques. And so that relates to the question that I asked myself: would AI be able to make of everybody a decent farmer? And it doesn't have to be restricted to crop science, of course, but this is something that intrigued me. So maybe Hugo, you could start if you have an opinion on this. It's a little bit outside our comfort zone of most of us, but still.
Well, to some extent, there are analogies. The field test is what for us the clinic is. Obviously, there are ethical restrictions on what you could experiment there. So we also have possibly even more extensive in vitro setting. But we see a very similar evolution there in terms of robotization, miniaturization, and evolution in technology, multiple readouts that go in there and that need to be processed. But there's then a difference between the use of that and can it make everyone into a good farmer? So on the one hand, you have the organizations, the departments, the people focusing on setting this up. On the other hand, you have the challenge of making that work relevant, translating those insights into supportive advice that can actually help the colleagues. So for me, the analogy pretty much holds. What is happening in agriculture is indeed also happening in the pharmacological setting. I don't think there is any company in the world that is not believing that this scaling is going to help. AI has a huge role in harnessing harnessing that. I think we already mentioned that in the previous discussion, harnessing those data and making those insights digestible for then the colleagues that will act on that will turn that into new products is and remains paramount.
Mm-hmm. Barbara?
Well, really, that's really also out of my comfort zone. So there's not so much to add, but I think, what pharma industry can make use of here is that really AI seems to work well in if you use image analysis, right? And that's what they're mainly doing. So they take pictures, and in this video also we saw that it seems to work, and it can really also help in the aspect of safety, right? And you can make farming more effective and reduce costs. So it might really help for smaller farms to reduce costs, but really hard for me to assess how large the impact will be. But I think it holds true what Hugo said, that probably it's a good analogy to drug discovery, that we have to see over the next couple of years what really the impact is, but it looks like especially image analysis is a very fruitful way to go forward here.
Maybe we can also extend it a little bit, like it's the intuition that I also remembered from this, so that a farmer thinks this is what has the impact, but maybe he has it wrong. And so maybe we can make the analogy to chemistry that we think already for many years, this is how this compound has to be made, and we are not open for any alternative anymore. So maybe AI can also take these, well, almost like false statements out of our equation, and maybe that we can be more, that the experience remains important, but at least we are open for alternatives suggested by AI, and maybe that's something, maybe Maarten also, from your perspective, that because you do a lot of inline censoring, and so that you sometimes see things that are counterintuitive, but definitely it's what the data says, right? So
yeah, so again, also agreeing with Hugo and Barbara, I think there's the basic question is, do you need an expert, or can you leave out the expert in the field, and can AI do the whole job in agriculture, and also, for example, in chemistry or in biology? And the answer, agreed, it's not. On the other hand, like you say, you can also use it to take away old ideas on how the world looks like, and how you should synthesize, and how you should manage. And also in industry, of course, many optimize processes on basis of temperature or pressure, or on their gut feeling, and they think that temperature was the driver, but actually it was not the only driver. And the correlation of, in this case, for example, physical parameters like viscosity, temperature, pressure, with molecular information, and not especially for the obvious reaction which you wanted to monitor, but the presence or absence of side products or runaway reactions or whatever, that will bring a lot of new insight. So yes, I agree, Tim, that it will help us to really dig into what are the real basics behind certain processes, and not only on our gut feeling. Although in many cases our gut feeling is based on a lot of actual experience, so it's a give and take there, I would say.
Maybe Ola, you could also tune in here, because you said that you're also interested in synthetic planning, and I can imagine that a lot of organic chemists says this is the way and no other way, but probably AI can give you a different route to the final compound.
So of course, the synthetic planning is important both in the agrochemistry as well as in pharma, but I think that there's also some differences that you need to include in your synthetic planning tool. So you need to have a flexible tool. For instance, cost of goods will be much more important in the agrochemistry compared to pharma, because you will do consider much more, and then that of course impacts that you should definitely use green chemistry and so on. So some other reflections I have. First, I'm actually I was at an EBI industry meeting, I think roughly almost 10 years ago now, about semantic technologies. And I was really impressed by the agro industry, how well already at that time they collected data, analyzed it, and semantically annotated the data. I think the pharma have catched up, but they were clearly much more earlier on the ball than the pharma industry. Another reflection, of course, I agree with all what the earlier speakers or panelists have said, is that I think ethics is really important. So I think for decision in the field, it can really impact, say, a farmer's economy. So both understanding of causality and also have real transparency in the system, I think is really paramount here.
Okay, great. Yeah, I think that settles this question as well, and so I think we can move on if you're all okay. If you don't have anything to add. So the next discussion was by Nathan Brown from Benevolent AI, and so he he said that the goal of AI is really to reduce time that it takes to bring a drug to the market. We already briefly touched upon that, and reduce the amount of drug attrition also in the development phase. In other words, make the discovery process much more efficient. So he was saying something about knowledge graphs built with all the published biomedical data, and that provides an unbiased overview of what has been published so far. So he also focuses on a mechanistic-based drug discovery. So every hit in silico is subsequently tested then to verify its predictions. And the advantage is that more work can be done in silico, and the amount of lab work can be reduced to what is actually needed. And so that saves a lot of time. So the statement to discuss here is that he says that in the near future, AI will be a driving, but not necessarily essential force to increase the effectiveness of drug development. So I think this touches upon pharmaceutical development. So I would like to start with Ola here again, if he has any ideas on the statement. Oh, you're still on.
Apologies. So basically, I think here, I mean, it's really what is essential here. And it's, I'm not 100% sure what Nathan is actually meaning with essential. But if you ask me what I think is the most essential, then it's not really related to AI and machine learning. I think the most essential is the data package that goes into the regulatory submissions, to the investigational new drug submission, to start a clinical trial, for the market approval after your phase 3 study. And that will always, I think, be based on solid experimental data. I don't think we will have a situation that you do a regulatory submission on AI predictions only and not experimental data. I don't think that ever will happen. So in essence, I think drug discovery and development is an experimental endeavor, and the key data that is needed for regulatory submission, which I consider to be the essential data, will be experimental. That said, of course, AI and machine learning can help tremendously in generating that data, to generate the right data much faster, much more efficient, hopefully in the future than historically.
Hugo, maybe from your vision from Janssen.
Yeah, so I agree with the statement that the package is all important, and to a large extent, to a full extent, there will need to be experimental data. However, I think we have all seen it takes an enormous amount of time and money, and that efficiency has to increase. And it is there where AI/ML should play its biggest role. With then respect, the first part of the question is critical, in my opinion, in the nearby future. What we always see is that we tend to overestimate what we can do in the next 3 years, and we tend to underestimate what we can do in the next 10 years. And the reason is fairly simple. In the next 3 years, we know all of the components, and we are a bit arrogant in, oh, we'll figure out how to combine them optimally. If we look at the next 10 years, there are going to be certain breakthroughs we don't anticipate, or we don't think that are going to make it. We saw that in the genomics era. We saw that in many areas. And I think AI/ML is going to benefit from that. So over the next 3 years, we see largely incremental. Yes, we speed up a bit here, we speed up a bit there. We bring that data upgrade, we harness more data. But there's going to be challenges in those that are going to make us move somewhat slower than we hope. At the same time, there will be some breakthroughs. I think some of the breakthrough, and I'm not going to go in extenso here because I think it will be covered later, is, for example, suddenly AlphaFold can do much better than ever before. If you would have asked 3 years ago, are we going there? I think, well, people have been saying that for decades. These things happen. It is these things, and then how we can integrate them with the rest of the tool package that are going to make a difference. So I think we've only seen the beginning. I think we tend to slightly overestimate what we'll be able to do in the next 2-3 years. But I think we hugely underestimate the role in coming to that experimental data package in a much more efficient way, that we underestimate that potential to this day.
Right, Barbara.
Okay, so I would have answered it a little bit different. Also, I really agree with the other panelists. So, because I was a bit, I didn't really understand why he would say in the near future. I think we are already seeing those developments, right? And if we define AI as more data-driven drug discovery, right, a bit broader, this is happening since 15 years now, right? We have this, or had this productivity crisis, still have it in pharmaceutical R&D. And we really have seen from the early beginnings of this century that data-driven approaches lead to a competitive advantage here, because diseases we are looking at are more complex now. It's also, we have to look at long-term safety effects. It's more chronic diseases, things like this. So really data-driven approaches are in the forefront, and without those, we wouldn't make any progress, I think. So this also has to be said, but it's also true that we can only, it's really hard to estimate what the impact of especially ML, deep learning, will be in 5 or 10 years. And I also have to second that what we are now seeing with AlphaFold 2 is amazing, and we can only start to think about what else is possible, but we will talk about this a bit later. So in my opinion, really, the data-driven approaches already have proven to be very important for drug discovery. And I can't really say where would we stand without those, right? After 2000, actually. So the digital transformation takes place already, of course, since 20 years, I think.
Maarten, from your hybrid experience.
Yeah, I was a little bit less triggered on nearby future, and I think I can, I'm not the person compared to the other panelists to say something about that, because I'm not having a vision there. I was much more triggered by the word effectiveness, and that means for me, first time right, or do something effectively, well, pre-thought, and so on. And I've been looking also to some Bayesian type of neural networking systems. You can compare it maybe with design of an experiments. In analytical chemistry, we have been using that a lot to minimize the number of experiments you have to do and be more effective to reach your goal. So is artificial intelligence or machine learning, deep learning, helping you to screen better and more focused, instead of just in the past screening millions of compounds against the target, which obviously was not necessary? And I absolutely believe so. I cannot anticipate how fast that field goes, and I agree that innovation processes go step by step, and then suddenly there is a new paradigm shift and it goes faster again. So for sure, the others have their better vision, but I absolutely believe that also the new algorithms and mathematical models to do Bayesian type neural networking, artificial networking, and deep learning will help us to be more effective in design and building and doing the experimentation in pharma. And but maybe Hugo can say something better there, because I'm out of pharma since 10 years for sure, but I absolutely see the added value. And also in analytical chemistry, that's maybe the last one, because I have looking to the literature, and yes, everything is related on data, on analytical data, but using artificial intelligence to increase effectiveness in design of analytical methodologies in pharma, I think that is still in its childhood, I would say. But Hugo, maybe you can say something better there.
So we are using data and many computational methods. To what extent they are AI ML doesn't even matter, because it's going to be updated and fused anyway. But many of those computational methods indeed do increase the efficiency. So you will get better hit rates. If you use structure-based virtual screening, you will get better hit rates. If you use image-based virtual screening, you will get better hit rates. If you use more models, you will get better hit rates. By combining them probabilistically with different methodology, you will improve efficiency. At the same time, we also have to observe that the targets are getting harder and harder. So it's an arms race. We are the easy targets that were still around 2 decades ago. Nowadays, you come with protein-protein interactions to tackle transcription factors, much more volatile targets where sometimes structure doesn't help. So it's a bit of an arms race. While the bar is being moved along, yes, our arsenal to address that gets better. Now, to remain a minimal of efficiency is going to be essential to the industry. If the cost becomes too high to put drugs on the market, that is going to scare away this endeavor. So that's why we should be alert. We should not be complacent. But on the other hand, we are seeing the signs that AI/ML indeed does prove to be a weapon, and in the broader sense, all computational methods do prove to be a method in raising the efficiency.
Mm-hmm. And it's maybe a question from me to you also is in the past, you were always looking what is the hit to the target. And Barbara was also referring to off target. So is the authorities now not only obliging you to explain how it works, but also why it does not work to other targets? And so they discuss selectivity. And that world is so complex and so big that again, that is a part of raising the bar for pharmaceutical industry to come up with new drugs.
So multiparametric is becoming more and more important. So where in the past you would have your primary activity, you would have 1 or 2 counter activities, and then perhaps a handful of run-of-the-mill, very, very frequent toxicology effects. We can see that remit is expanding. The field is more and more aware of what can go wrong. Specificity is indeed important. So people are trying to come up with methods. Now, obviously, on the one hand, the question is, do we have all the data that is necessary? For some aspects, that is the case. And you can deploy data generation to improve those methods. Data generation both in classical methods, but also in newer methodologies. Barbara mentioned imaging, and it's a fantastic tool for many of the off targets. Whatever stresses a cell will, to some extent, translate into morphology. But it is indeed, you need not just the image, you need not just the compounds, you also need the annotation. What does this do? How do we interpret this? And how can we control the costs? Because ultimately, the more of those things you start exploring, the more it costs. We will increase the efficiency. The bar is getting higher. You need to balance that. But you're absolutely right. The bar is rising also there. People are more aware and are expecting you to cast a broader safety net. So at least the list of things you should avoid is slowly, but very certainly extending. And so also the sensors we have in place to flag for those things happening. And then secondly, the tools to move away from those once you're flagged definitely are high up the radar. And I'm convinced of many pharma, but definitely we're paying close attention to that.
Maybe I can add, I don't think there's any regulatory need to submit off-target data, except for HERG. That is a regulatory requirement. But in the end, you want to maximize efficacy as the lowest possible dose. So there is, of course, a lot of investigation of potential off-targets, both prediction and experimental work. To be able to minimize off-target effects, so you can actually maximize the efficacy as the lowest possible dose.
Okay. Anybody else wants to add something still?
Maybe one thing that also
Yes.
Nick mentioned in his talk was about, okay, he talked about knowledge graphs they're using at Benevolent, and that they really use all this knowledge for already at the stage of target identification. And I think this fits well into this discussion and also with off targets. So in the end, I think also a big gain here now is that with all this data and when we connect it, we can already at the stage of identifying the targets that we want to work on, have more informed decisions maybe rather than later where we then suddenly realize, okay, there are off-target effects. So really, yeah, gathering all this information as an early stage enables us to take more informed decisions, and maybe this will be one of the main breakthroughs, or is already there. I don't know because I'm not in pharma, but I think that's something he really mentioned in his talk. It's a very informed target identification procedure, but always all coupled to experimental testing, of course. Just wanted to add this. Thanks.
Oh, thank you. If that is all, we can move on to the next point of discussion, which was by Christoph Steinbeck from Friedrich Schiller University. And so his main question that he had was, do we have enough data in chemistry, in academic chemistry, to use deep learning? And so for that, data must be open source and downloadable. And we have really a culture problem. I think we touched upon that already in one of the previous discussions, that we have a culture problem in chemistry. ELNs are very rare in academia. And so we need to make our data much more findable, accessible, interoperable, and reusable. And so once everybody does that, there will be plenty of data that can be used for machine learning. And so he says all those handwritten notebooks, and I think in one of the earlier discussions, I think you, Maarten, said that there were notebooks full of data, but they are actually very difficult to read by any machines. And so how do you liberate the data, that old data, and digitize it? So this is definitely, I think, a discussion about digitization of chemistry. And so the statement that that was launched for that associated with this talk from Christoph Steinbeck was, why do chemists like their handwritten lab journals so much, and how can we move them to digital? I think in the industry, it's only ELN, right? So maybe we can start here with Maarten from academia. He has seen both sides, and maybe you have an opinion on this.
Yeah. Tim, yeah, I don't see any ELN at universities, and even I saw at certain R&D in industry no ELN. But when I was in pharma, for sure, and I was there in the beginning of, at the end of the 90s. And as an analytical lab, I had in our archive all the data, all the analytical data. And actually, we had a 2-year project then in digitalization of all the paper data into a PDF format, and already searchable. So we could search for words in these PDFs. And I don't know where the technology is nowadays, but I presume that there's technologies who can search PDF files for words and recognition. Yesterday I was looking to my son, who was writing on such a keynote notebook, and with his handwriting, the machine already knew it and wrote it nice in typed letters. So there should be some technology. That's first of all. I don't know why people want to stay with paperwork. I don't have an idea there. And if you look to ELN, it is easy, and it's also retrievable, easily retrievable to do so. So you would go from looking back in your lab books, not knowing which number, to very fast retrievable data in your ELN. And another thing is, and that was particular, especially in discovery, important, is that ELN was not only there to archive all the information, but also take care that you can witness on the time of invent of a chemical. So patenting and the time and originality of a finding was even more important in the ELN at drug discovery departments than in drug development. So I did not give really an answer to your question, why is nobody going to ELN? I don't know. This is maybe, again, such a behavior of not understanding yet what technology can bring you. And then when you know it, then suddenly you are happy that it brought a lot of things. My experience was in that transition in pharma from the 90s to let's say the beginning of the 2000s. It helped us a lot, and especially my analytical chemists, by quickly retrieving methodologies which were done or developed years before, which we a little bit forgot or misinterpreted over time.
Barbara, maybe
Yeah, so I also don't have a conclusive answer. I also think it's a bad old habit, basically. And maybe in chemistry, really, since we want to draw chemical structures, sometimes that's why we do it by hand. But so in my lab, we do it electronically, I have to say, but really basically because it's searchable. And if somebody leaves, it's much easier to just then hand it over. And we have seen with old lab books that are handwritten that over time also after some years, you can't read them anymore, right? Depending on what pen you used. So we do it electronically. I really also think it's just this old habit. And what is also important in the context of this discussion is, I think that was mentioned in the talk, is now that all the data is in papers, right? How we can better make use of this data that is also in papers in scientific literature. Because that's, okay, electronic, but it's PDF. So how do we extract the data? And I think one step forward would really be, as I mentioned before, that we try to convince the community that we need chemical structures in a machine-readable format, for instance. So, or as we do it for protein structures that are submitted to PDB, of course, we could also submit chemical structures to PubChem, for instance, so to make that easier. But not to forget, in literature, there is a wealth of data that we should make use of, and that was mentioned in the talk as well. So we can use all this historical data to make informed decisions. That's one of my favorite topics also in my research, to look at trends in chemical data over time. And it's easier with databases like Campbell, for instance, but there are journals that aren't covered. So, I really think, and that was really mentioned, thinking about the future, we should make it easier for the community to retrieve the data, not only within the labs, but also for the wider community when we publish. Think about solutions so that data can be accessed in an easier way. But as I said, I really think it's just a bad habit, basically, why people just write by hand. And I guess these notebooks can manage chemical structures, right? We seldomly write them down, but I think they can manage that you draw them, right? So that's not a limitation.
Maybe now the industry perspective, Hugo.
So first of all, the fight between handwritten and electronic, I think that was fought long before I even joined, and there's not even a question on that. This being said, that is not the end of it. There is a cost to have a chemist annotate and the level of detail to which he annotates. And then finally, the protocols. Can we make the protocols more uniform so that it's consistent? So if we go back even to electronic data, it's a massive advantage over handwritten, but even there are gaps. We see a lot of promise there from automated chemistry, not just as an execution tool, but also as a tool. Per definition, if you have a robot execute a reaction, every single part of that reaction, every condition, every sensor readout is there in a digital format. As we harness those data, one of the challenges is, can we make sure that we level up? So that is the gold standard, so very detailed. We level up to that rather than stripping the information out and bringing that bar to what a human would insert on that. We should be smart if we're going to ask our chemists to make that annotation to such a level of detail that it's competitive with what a machine could digitally transfer. That would be unfair. They are doing their best to capture that, but there is a certain economy. So I also think that there still is an outstanding challenge to make things like auto-completion, so pre-populate certain things so that we use the time of the chemist as sparingly as possible. Is it a problem that has been solved? Not yet completely. So part of the solutions are there, but if we are looking at either commercial or internal databases or patent-based databases that are publicly available, all of them contain a lot of invalid reactions. Even if you just do the balancing out, even if you do inferences, there is a lot of fuzziness in this data. But we do think that automation, that supporting chemists, manual chemists, to do this job much more efficiently, with auto checks in place at the time of recording, that those can go a long way. But luckily, at least from an industry perspective, we are not competing with handwritten anymore. That discussion has been closed a long time ago.
Ola.
Okay, yes. So I think I agree a lot with what Hugo already said. I think if you look at the electronic lab notebooks, they are doing very well for what they're actually designed for, to capture IP. But unfortunately, they are quite unstructured, so to say, in their data. There's a lot of comments. Chemists, for instance, can themselves add which catalyst they've been using. It's not like potentially in a drop-down menu, which means that the chemist can write the catalyst in many different ways, which then is different to disentangle when you actually try to automatically extract the data. So it's so definitely, I think there should be more work ongoing, also actually in collaboration with the ELN vendors to have much more structured ELN notebooks going forward than we had historically. I think there's more data also to be captured, as Hugo also mentioned. I mean, we are moving in more and more into high throughput chemistry. I think there's still definitely some limitations there. But it's clearly direction of the field with robotics, and there are, for instance, startups also like Deep Matter coming out from Lee Cronin's lab, try to capture the data much more accurate actually during the reaction. So there's a lot of things going on, but I think it's with the so to say with the statement, there's one more thing that I think is important, and that's, so to say, the chemists and all scientists. I think there's a question: what's in it for them to annotate the data better? That could be for the chemists and biologists. So I think it's important, both in academia and in industry, to somehow have some type of reward system that actually, if you annotate your data well, not only you can reproduce the results, but it's stored in a proper way. There is a reward system in some way or the other. It probably can be discussed how you should reward, but I think that the data generator, and particularly when they annotate the data well, needs really to be acknowledged for the great work they are doing, because that's in the end essential for machine learning and AI. And that reward part, I think, is very important. So to encourage people to better annotate their data, both chemists, biologists, pharmacologists, and so on.
Mm-hmm. Maybe I can add something about that. The reward would be understanding on what you're doing, and understanding the basics and the fundamentals of science, I would say. And we have been discussing that on the necessity of experts and experts on annotation. And maybe it's an old schoenendoos, we say in Dutch, discussion, but in the 90s, I had two technicians doing Raman and infrared spectroscopy on crystallization processes, especially in polymorphs of steroids. And sometimes we had mixtures of 6 or 7, sometimes even unstable polymorphs. And they could see in a split second on a Raman spectrum infrared and classify the different polymorphs. And we tried that with conventional PCA and PLS chemomatics, and we were not able to do so. Because and they could even translate experimental conditions to the quality of the spectra and how they could distinguish certain polymorphs. And likewise, we have been fighting in that time for a year to time warp or to calibrate NMR data in such a manner that we had readable data to support metabolomics and biological output there. So where will artificial intelligence and machine learning, deep learning help us is to understand the correlation between experimental conditions and the quality of the data is the trueness and the information power of a data point. And I think that is also important. And in all those times, the analytical scientists working in my group were linking structures to Fischem properties and metabolic stability. And they became able on not only perform the analysis, but also understanding if the analysis was right on the basis what the findings were. So the award could be, as a proposal, Ola, is that it will help to annotate better and better understand what the hell we are doing in drug discovery and drug development.
But I think the reward could also be citations, right? Because we all want papers and citations. So if we would, for instance, deposit our data in a repository and this data would get a DOI and then it could be cited by others, this could be a reward system, but not sure if that would work, right? Because it's still a lot of effort to annotate the data, and I'm not sure that's enough award or reward for everybody. And also it would again support this system of citations and impact factors, and I'm not sure that's the best system to have.
From an academic point of view, that should be highly questionable.
Exactly.
Okay. I think we handled this question quite well so far. So then we are already going to our last one. So if people have any questions, please do not forget to use the Q&A box and raise some questions. We already dealt with 2, but there is, of course, much more room for other questions. So so please do use the button, and we can then handle them live in this session. So the last talk that we had was from Lucy Colwell from Google Research and University of Cambridge. And so she said that protein sequence modeling, and that's also what somebody said already in the Q&A, it's a great field to work on because of the huge amount of data that is open access available. And we also briefly touched about that in one of our previous discussions. Now, protein structures can be predicted. AlphaFold was already mentioned, which is amazing. I think we didn't see that. Well, it was predicted that it could be done, but all of a sudden, the precision of AlphaFold was phenomenal. And so from Dr. Colwell's lecture, we can also learn that it might be possible to even link that to the function of those proteins. And so the statement to discuss that she raised was that DeepMind technology and AlphaFold will eventually be able to not only predict protein structures 100% accurately, but also predict the function of those proteins with high precision. So maybe that's something that I would like to throw first at Ola and Hugo. So maybe Ola, you can kick off. Do you think, how far are we from, well, AlphaFold already showed that we can predict protein structures. So how far are we from the function?
Yeah, of course, me, like everyone else, is really both impressed and enthusiastic about the progress with AlphaFold 2. It's really impressive. The Nature paper is probably one of the best computational paper, at least in life sciences, out there. Really impressive work. But that said, I mean, structure is clearly not everything, and of course that's their knowledge as well. And then it comes from structure. That's basically one snapshot, but you have the dynamics. Some proteins are very ordered. Then, of course, one snapshot is perfectly fine. Other proteins are much more disordered. And then just one snapshot is not saying that much because it's really a huge ensemble of different structure present, depending on conditions and so on. So I think it's the next step is to really understand the dynamics. And of course, that can be done also with computational methods like molecular dynamics. Experimental method will for sure add as well, like NMR. And then you come to function. Of course, sometimes for function, you could actually predict it from the sequence. I mean, you can see if it's a GPCR or protease and so on. So actually quite a lot of core functions you know already from the sequence. You don't need a 3D structure. I think the 3D structure can add more, definitely functional information, particular binding partner. And there's not only AlphaFold, I think RosettaFold, which is similar from David Baker's lab, is working a lot to better predict protein-protein interactions. I think that will be the key to better understand the function. I think there will be a lot of progress there. But I think it's not only will be computational. I think there will be also in experiments a lot of progress that will feed into to improve the computational method of protein-protein interaction. I'm thinking about cryo-EM, where suddenly you can crystallize much more bigger complexes that you can done historically, and you can potentially also get information from proteomics. So I think there will be a lot of progress. If you can capture all types of function, I'm a bit skeptical because it can be pretty complex. You might have post-translational modifications that then will change the function and so on. And also what I think is important is that you only predict the potential function. If that function will actually take place, it depends which partners are expressed in the same cellular compartment. Because a function can only take place if all partners necessarily are expressed, and there's no other competing partner that is also expressed that might have higher affinity. So in the end, you will only predict potential functions. If it happens or not will depend a lot of the context in the cellular compartment.
Right. I agree with that. Hugo.
Ola covered already a lot of ground. So first of all, AlphaFold and RosettaFold, indeed fantastic achievements, a breakthrough in a challenge that has mesmerized a whole generation of scientists. This being said, also like Ola already hinted at, it focuses very much on a biochemical view. I'm reducing my disease to a target or a handful of targets. You're not looking at the dynamics. AI/ML is going to have a massive impact on the acceleration of a lot of the molecular dynamics technology that exists. So not necessarily changing the quality of that, but changing the speed. And hence the volume of structures, the time tracks that become possible. But still, I have a bit of a hard time to see how that will bridge. I'm very hopeful, and as a data scientist, I'm very enthusiastic about what progress can bring. But I have a bit of a hard time. For me, function ultimately of a drug means what does it do in a patient? And I think the structural resolution, however great this breakthrough was, the structural resolution that we have achieved now is only one of the very early steps. We are nowhere near the temporal resolution, the complexity in a cellular context, then the lifting that complexity to the scale of organs, to the scale of what an actual human being does. So it all depends. If you're talking in a purely biochemical way, I can recognize a certain domain, or I can recognize a certain regulatory aspect of that domain. Yes, then we could concur with that statement. But if you put the bar higher, will it explain what the function of that protein will do in a patient context? Then I think it is only it is one step. It is only one angle, a very important angle, but it's only one angle. I think a big value of AI/ML is providing the ability of looking at multiple views on the same process. So this is very much focused on the biochemical, structure-based, so essentially helping with predicting biochemical readouts, enzyme kinetic readouts, things like that, assays that are relatively close to a biochemical system. But Barbara already mentioned, and we also have done quite some work on that, the value of microscopy images to demonstrate biology, at least at a cellular level, and possibly in the nearby future. And another observation is that even if I today talk to the CAT colleagues who do the structure-based virtual screen, they always ask us for, oh, can we have a look at the actual hits to calibrate the methods? So even though those methods are very powerful, you need calibration, you need some ground truth. And that ground truth comes from the assays. even the biochemical assays, but then let alone if those assays become more complex. So while I completely concur, fantastic breakthrough, and I sincerely hope that this work can be continued, and I hope this will have a return. I do not agree with that this is the answer, this is the panacea that's going to solve it all. We have to, at the same time, also invest in complementary methods, image methods, other molecular methods, sequencing methods, all kinds of electrophysiological readouts. The broader the set of glasses, the set of angles is that we can throw at this, the more likely we are to make headway and put better and safer drugs in patients.
Precision of the data is crucial here, and actually analyzing everything in the proper way is key.
It's not just the precision of data. The question is very much honed to structure is everything. This is what, in principle, we can explain everything from the 3 major physical laws. But we know we're not there. We can't do that. We need a lot of calibration. We need to distill that down to things that are more manageable. And that is where all these different angles help. So while in theory, yes, with the physical and chemophysical information and the laws of physics, you could deduce it all. In practice, I don't think that is the most efficient way, that this is the fastest way to help our patients.
Okay. Barbara?
Yeah, so I can't agree more with what Ola and Hugo already said in terms of what a tremendous breakthrough this is for all of us, and how we will certainly make use of that, but also about the limitations. And I agree that probably function you can't really determine 100%, but it will bring us closer towards an understanding from a structural perspective. And especially I see the advantage where you want to study whole protein families now, for instance. So you can also look at what mutations, because you can, the code is all available, you can introduce mutations and then see what effect has this on the structure, for instance. And I think this will really give a boost to research fields where targets are understudied, especially in my domain, transporters, for instance. We didn't have a lot of structures, or we don't still have a lot of experimental structures. Now with cryo-EM, these last few years, there were a few more structures, but still, especially for my targets, the organic anion transporting polypeptides, we didn't have a structure and not from any species of this family. So what we did is we used the threading approach, which also includes an SVM model. But then what we in addition did, and this refers to the point that Ola mentioned, and I think that's really important, the dynamics. So even though we used the threading approach and we built a model, what we did is we used normal mode analysis to really sample conformations. And then we found out, and then we validated this by an enrichment docking procedure. And we found out that the best model basically wasn't the starting template, but a different conformation. So this basically will not be solved, right, by AlphaFold 2 now. But really, I think, so now I started to actually compare their structures to our structures. And I think in the transporter field, for instance, or also for neglected diseases, rare diseases, where probably, in fields where there are not many experimental structures, I think that this will lead to a boost in research, but we have to understand the limitations. And they were already lined out by my colleagues here now. So intrinsically disordered regions of proteins, conformational changes are important. Post-translational modifications can't be captured yet. Protein-protein interactions. So with all these limitations, still, I think it's very great, and we will see in the next few years where to get with these methods.
Maarten.
Yeah, I absolutely agree. I already introduced myself as being focused on dynamic systems, so I don't believe in just a structure. It's the dynamics and the conformation of molecules which do some energetic work. And my old boss in medicinal chemistry always asked me, you are an analytical chemist, so please can you analyze why a protein at time 0 is a protein and 1 picosecond later suddenly it's a substrate for a protease? And can you measure that, and can you explain me why that's happening? And of course that has to do something with the basics of science in terms of energy and conformational changes. And especially also in the context. And I think that is very important what Hugo has been saying. Some of my colleagues said we don't use in vitro assays, or not even in vivo, not in vitro cellulary assays, but we just go into vivo because that's giving us the answer if it works or not works in the end. And another interesting thing is that we have been developing, of course, apart from electron microscopy, which I regard as really a big step forward, a lot of X-ray crystallography and NMR methodologies. And we will apply high field NMR for molecular dynamics there also. And in combination with this type of information, of course, we can start to give a little bit light. Not only saying that analytical scientists with whatever type of machine are not really getting a structure which is called native. And in mass spectrometry, and I'm a mass spectrometrist, there's a lot of effort in analyzing the native structure of proteins, and we don't actually know what is the native structure because we cannot see in the cell, like Barbara says, or in the cell membrane in time and what's going on. So imaging, mass spectrometry imaging, electron microscopy imaging and I would even say atomic scale is becoming there and together. And I absolutely believe, I've seen pictures in our own institute by Peter Peters, where they have on an atomic level identified very complex protein complexes. But again, the technologies, AlphaFold and the analytical technology will force it, but dynamics is still far away, I would say. But hopefully we will come there one day.
Okay. Anybody wants to add something to this discussion?
Yeah, I think I just want to reflect a little bit on what Hugo said, because I think he raised an interesting point, which I think is a discussion point within each pharma company. It's around a more holistic view versus a very reductionistic view. And I think that there's clearly different schools there. And if I understand Hugo, he would like to more see drug design from a holistic perspective, where more advanced assays are coming in, like cell paint and so on. And I think that's a school I personally agree with. But of course, there's another school that is very reductionistic. And for them, of course, the structure is very important. And that school is basically relying a lot on the human genetic validation to see that as the core. So say if you have a GPCR, you have mutations in the GPCRs, say related to obesity, take the simple example, that some mutation related to increased weight, some other mutation is increases to weight loss. Then of course you have a very strong reductionistic validation, and then many would argue that you will actually focus on a reductionistic perspective, where the structure will be a key piece, and so to say, and a more holistic perspective would be less relevant. But that I think is a debatable point within all pharma, and I personally think a holistic perspective have a lot of advantages.
Hugo.
It's probably not black and white. I like structure based as much as the next guy, and it has brought tremendous value. On the other hand, we also see a bit of a low hanging fruit phenomenon, where the most straightforward cases where we were lucky enough that a certain set of simple mutations, so there was a fairly simple correlation between some set of mutations and a strong separation of a clinical phenotype. I am afraid that most of us around this table are very much aware that in the vast cases, the mutation field is much more complex. There's lots of confounding factors. So having those smoking guns, wherever they existed, they have been targeted for a while. and wherever they were druggable, some of them may already have made it, some of them are still looked into. As long as that yields good efficiency, there's nothing against that. On the other hand, we know that that list is getting shorter and shorter. And we know that there are still a lot of unmet medical needs we can possibly not address with that approach. And so that's the reason why would we not avail ourselves of an arsenal of tools where we can deploy and whatever works, and you can take the easiest, the most straightforward, the best established first. But the second you start realizing I'm not getting the full picture, complement that with additional approaches. For some of the hard targets, we see that many companies, also many academic groups, start realizing single modality is not the only answer. So going multimodality searches, complementing traditional screens with DEL modalities, complementing biochemical assays with cellular assays in different shapes. We see this more and more. We still have to care about efficiency. And I think that is the main argument. We should not go, the sky is the limit. We throw everything, the kitchen sink at every single stage. So we should be smart. How do we stage that the most efficiently? And I think at in different constellations, you may come to a different conclusion. But as with most things in life, the extremes of the spectrum are probably not the place where you want to be. You want to settle somewhere closer to that sweet spot, which typically is closer in the middle.
I agree there. I agree. Just wanted to point out the different philosophies. Yeah.
Absolutely.
Anything else to add? Actually, we went through all the questions a bit faster than we anticipated, but I think also some questions were, or some statements were very much linked together. So you cannot extend the discussion much longer without being redundant, I would say. Maybe one question that I would like to throw in there for everybody is like, if we already said we tend to over-predict in 3 years and under-predict in 10 years, but if there is something for your own field that you would like to see really solved, what would be it? And maybe that's a hard question, but yeah, if you have something in mind that you think is a grand challenge and that probably we will be able to fix in the next decade or so, what would be it? So yeah, Maarten, maybe you can start.
No, a very important one, I think. I'm a mass spectrometrist, and I've been working for nearly 40 years now with mass spectrometry, and I've seen mass spectrometry in metabolomics in all this omics type of science. And the interfaces and the technologies used are normally electrospray or MALDI ionization processes in mass spectrometry. And I can guarantee you the most understood or non-understood ionization process and the most complex ones are electrospray and MALDI. And sometimes I've been saying that I can give you a sample and you only will see 5% of all the molecules which are there. And then when you see them, the relative ratio between these molecules is per definition not right. So we are entering all this data into metabolomics, and then we try to do some Hersky-Mersky and then say, okay, we see a correlation, which is still far off causality. Now, this is a whole story. So I'm very intrigued, and I'm absolutely sure I would even say that artificial neural networks and machine and deep learning will help me to unravel the basic processes behind ionization processes in mass spectrometry and helping to predict, to make better experimental conditions to have a much better data output into the data of metabolomics. And I think this is an unmet need. There is a lot published, I would say sometimes a lot published, which is not incorrect, but incomplete, I would say. And there we have a big target. So I would definitely like to invite my colleagues in artificial intelligence and machine learning and deep learning to help me out to understand ionization processes in electrospray and MALDI.
Thank you. Hugo.
Or there's a whole list of things that could be the next breakthrough. One of the things we're very enthusiastic about is synthesis reaction. I think the main challenge there is data. We've covered that. So predicting that, while a lot of data exists, a lot of data has not been captured or has been captured in an incomplete way. But what we have seen is that with the data that is available, if we compare this field with where it was 3 to 4 years ago, a massive jump has been made. Are we there? No. So for me, the biggest challenge, but then also the hope that could have a big impact is how can we become better at tapping into the data that is needed to learn that better. So that's definitely one of the things. A second question to address is how can we come up with additional ways of learning from one another in a non-naive way. We are currently engaged in a project that's called Melody with 10 different pharma partners. AstraZeneca, that is represented by Ola on this table, is one of those partners there. It is only one incarnation. We are seeing some signs of transfer. We obviously hope for that the sky is the limit there, but how can we untap that type of data? Thirdly, the image space. We see that what we can learn from images and also the pace at which images can be generated is picking up in the field. We also see startups valorizing that. So that is a third avenue where we see a lot of potential. I don't think we have the killer app, the definitive proof. But those are three areas where I think a combination of data challenges to solve, but it seems that this is possible. And on the other hand, evolving methodologies are very, very promising that one, two, or even three of those could actually become some of those breakthroughs, like the one from AlphaFold, that may enable a speed up towards increased efficiency.
Thank you. Barbara.
Yes, so it's quite interesting. If you would have asked me this question 5 years ago, I would have said I want some crystal structures for transporters, but I think now my vision has changed somehow. So I'm also more interested in more holistic approaches, especially I'm interested in what can we learn out of the historical data, right? That is in literature or in different data sources. So structured and unstructured data, right? We have a lot of data in patents, clinical data, databases like Campbell, and then in papers. And we tried to do this a little bit by looking into scaffold trends over time. Then we looked into target trends. So what I really would like to develop is really a system that enables us to take better informed decisions in the future. What is the target to focus on? What disease should we focus on? Things like this. And this, I think, includes a lot of different aspects. On one hand, it includes factors like druggability of a target, safety aspects of a target. So target safety is really insufficiently understood, I think. But it also includes things that you can extract from literature, for instance, which reflect the attention that the community pays to a target. And this can also help because it can basically show us where's the center of attention within a target family, where do many groups already work on, and maybe then I don't want to work on this as well, or was there a transition from preclinical to clinical state and so on. So there are a lot of limitations still to do such a global analysis, especially data, right? Again, so I'm not convinced that the openly available data, the publicly available data is enough to develop such a tool. But this, so I don't think that a lot of new technologies are needed to do that, but mainly data and needs to be standardized and made available to be able to do that. So this would be one of my visions, to really base our decisions in academia, but also in pharma, more on these critical factors that are really based on what we already know from historical data.
Ola, what's on your wish list?
Okay, so I think Barbara, Hugo, and Maarten brought us a lot of very good points already, but I would like to focus actually on something else. And that's actually, I think we have done a lot of progress in machine learning, AI, and I think the bottlenecks, if you look particularly what I'm doing around drug design, have moved to other parts of the value chain. One example, I think, is chemistry automation for discovery chemistry, still in pretty early stage. I think in process chemistry, it's much better to optimize the reaction, but to have a flexible system for discovery chemistry, if you want to go beyond one-step reaction, if you want to do multi-step reaction with the intermediate purification, I don't think anyone can do that routinely on a broad chemistry basis. There was one famous professor that told me that he could do it, but it turned out he could only do it as long as you want to do a Suzuki coupling, and that's not always what you want to do. So I think the flexibility there for automation discovery chemistry is still early on, and I think if you want to really cut timelines, there need to be progress there.
So data generator.
Yes. So, and the second part is, I mean, when we generate molecules and score them, I think the biggest bottleneck is affinity prediction. And I think if you're serious, you can do pretty well. But say if you want to find an initial hit to do scaffold hopping, then the methods are not really good yet. So I think techniques like absolute binding free energy, the development there, and of course a lot of groups are working on that, and potentially also combining with machine learning, AI, that would have a big breakthrough what we're doing if we can better estimate the affinity for novel scaffolds or for initial hits where no active compounds are known.
Thank you. In the meanwhile that we were discussing this final question, there came actually a question from the audience, from Vishnu Burman. And he said, like, can data-driven approaches create new and creative target molecules in drug discovery? This was in the past attributed to serendipity and strokes of luck, and thinking outside of the box, call it like that. So do you think AI machine learning will be able to increase this, stroke of luck? And maybe we can start with you, Ola, again, if you.
So I interpret the question is that with target molecules, so to say, Vishnu means small molecules that you basically want to design. First, you generate IDs and then you score them and synthesize them. And I think there should be creativity there, but it should be focused creativity. So it should not be too much extrapolation. I think the generation of new molecules with AI should stay in the space where you have trained on. So if you have trained on synthetically accessible molecules, the network should not go really creative and start to generate molecules that is very difficult to synthesize. So I believe in creativity there, but it should be restrained.
Yeah. Hugo, maybe
it sounds a bit like when generative AI was very new. The idea was, can we sample this one molecule? It may be a bit far out, but can we sample this one molecule? We make it, and we have a high enough probability that it is spot on. I think in the meantime, I mean, some people have invested time in this. I think we have to be humble. Our models are not good enough to do that. To just pinpoint, this is the one, it's far out from everything else. Like Ola says, extrapolation. On the other hand, with the increasing sets of data, we can make certain jumps that are not immediately intuitive. Is that creativity? Well, if an automation can do it, it always becomes almost philosophical whether that is still creative if you can code it. But it definitely is unintuitive. So certain modifications can be proposed where there seems to be a lot of evidence in the data that there is a high enough probability. It's not just a singleton, it is carried, it is synthesizable. So it is in the remit of the possible. But it never is as easy as completely blind, going out in the universe and somewhere in that spectrum, point to one spot, make that, and it's gonna solve everything. I think we're very, very far from that. But we are getting more efficient in exploring our solar system. Let's put it this way, where we know the planets a bit, where we have sent some probes out, we know the behavior, and yes, we are still bumping into unintuitive finds there. And so there